Here we are. So good afternoon. First of all, we really like to thank the organizers for inviting us to this webinar series. We're very happy to be able to be joining you today. As Alfred just introduced, my name is Tatiana Tiago, and I'm the product manager at Creoptix. So the second session of this webinar, it's really going to be split into two parts as shown here. So the first part will be presented by myself, and I'll be discussing the challenges and opportunities in drug screening. So really making an introduction as well to you of the Creoptix WAVEsystem and our GCI technology. The second part will be presented by our partners from Domainex. So Dr. Tom Mander, CEO at Domainex, will introduce the CRO and how they're supporting the drug discovery process, followed by Dr. Trevor Askwith, Head of Assay Biology at Domainex, will then share how the GCI technology and the WAVEsystem is helping his team really tackle fragment library screening. So we would also very much like to thank Domainex for joining us today. So then I'll start with the second session of the webinar, presenting to you, as I mentioned, the challenges and opportunities in drug discovery and giving you an introduction from Creoptix. So first, I would like to introduce a little bit who we are, then really explaining what grating coupled interferometry or GCI is and how this can address some of the challenges faced in drug discovery. And then doing an introduction to our latest technology introduced to waveRAPID, which is really a new way of measuring kinetics. So Creoptix is headquartered in Wädenswil at the edge of the Lake of Zurich in Switzerland. And here you can see a lovely picture from Wädenswil. You can really see what a nice location it is for us to be based. And our mission really is to enable life scientists to accelerate discovery by providing cutting-edge tools for molecular interaction analysis. And it's really focusing on the drug discovery markets and the needs of this market. So our own label-free optical biosensor, the WAVEsystem, is based on the proprietary microfluidics and our own grating-coupled interferometry or GCI technology. Outside of HQ in Switzerland, we count in US offices in the Boston area, and we have employees based in Germany, UK, and Sweden as well. Okay, so this is what our product looks like, the WAVEsystem. And it's really composed of a few different components. So one of the first and probably one of the main ones is, of course, the wave core. So this is really the biosensor unit featuring the GCI technology for detection, which really provides very high sensitivity and high signal to noise ratio. And this is coupled then to the wave sampler, which is a temperature controlled autosampler with capacity to 296 or 384 well plates. The wave controlled, which is an intuitive software that allows to operate the device. And this also includes our novel kinetic methods, the waveRAPID that I will introduce to you a little bit later, as well as the evidence-based data evaluation tool, Direct Kinetics, that really allows to not only analyze, but really provide the data much faster and with a lot less bias as well. Furthermore, we also have the wave chips, which are not shown in this image here, but these are the patented microfluidic cartridges that are compatible with a variety of samples, including crude samples, as well as samples containing harsh solvents. So now I'd really like to explain a little bit more what the technology, the GCI technology is, so the technology we're based on, and how really this is correlating and helping really overcome the drug discovery challenges. So there are several pain points in the drug discovery, as you're most likely aware. So one of the main ones is that it really takes weeks to get binding data, so it's really considerably slowing down the process from heat to lead screening. Often the data obtained using traditional techniques offers limited information. So here, often it only provides affinity data rather than the full kinetic characterization. So this is association and dissociation rates. Experts are often required to analyze, interpret the data when using this more complex technologies, which is really also slowing down the process in terms of the evaluation and the decision making. And when working with crude samples, so that really means, for example, unpurified samples, samples containing serum or cell lysates, for example, it often leads to clogging of the instrumentation, which is consequently leading to downtime and, again, really slowing all the discovery process. And often as well, it happens that the existing solutions on the market that are able to measure in higher throughput, they often do so at the expense of the sensitivity, really lowering the quality of the data. So sometimes to get the higher throughputs, really quality has to be a bit compromised. So all of these lead to bottlenecks in the drug discovery workflow, making it a long process, and therefore speeding up the early stages of drug discovery is really crucial to getting new medicines to patients faster. So over the years, label-free detection is gaining quite a lot widespread acceptance as an important research and development tool, as it analyzes molecular interactions without the need for additional isotope or fluorescent labels. So as we can see here, in traditional techniques, often it requires an individual bioassay component to be labeled. So often a secondary antibody, for example, or a secondary, for example, to be able to measure the binding event. And this often leads to distorted binding. On the other hand, label-free technology uses impedance or optic-based biosensor to measure the changes that occur directly upon binding. So as you can see here, the measurement here done by the optical field really is with a direct binding event without the need of secondary labels. So this really means that there is reduced non-specific binding and reduced background signal due to the not having the extra labels as well added to the complex. These are real-time results. It means that the measurement is done as it's happening. So as the binding is happening, the measurement is taking place. And this really allows to have the full kinetic characterization, so the association and dissociation time. So allowing also to measure not only if there is binding event, but how this binding is really behaving. Then the artificial manipulation of binding partners are not required, which ultimately leads to more physiologically relevant molecular interaction data. So SPR, BLI, and GCI are all label-free biosensor technologies that focus on the analysis of molecular interaction. So these optical biosensory technologies all measure changes in the refractive index close to a sensor surface, as shown in the pictures here. However, they are completely different technologies. So while SPR, for example, is based on a localized field here, and BLI is often affected by the diffusion-limited conditions due to the absence of microfluidics, GCI has a larger sensing area, as we can see here, which really means that it's seeing more interactions. So ultimately, this allows for more binding events to contribute to the overall signal that is detected here, giving GCI an intrinsically higher sensitivity to the label-free interaction. But now let me explain a little bit more really how GCI works. So as mentioned before, GCI is an optical biosensing technology that measures changes in the refractive index close to a sensor surface, as shown before. So it's based on the waveguide interferometry, which creates an evanescent field that spans the entire length of the sensor surface, as shown here. However, the advantage of our GCI readout is that the interferogram is created within the waveguide instead of being parallel to the waveguide as in traditional waveguide interferometry. So this really allows for a more robust readout compared to the classical waveguide interferometry, which it's regardless of temperature drifts or vibrations, which really translates into a much higher superior resolution over time. So also coupled with our GCI technology, as I mentioned previously, Creoptix uses microfluidic chips as shown here. So they were not shown in that initial image, but you can see here representation. And these chips do not have integral membranes. So this really eliminates the clogging issues that often occurs also with these devices. So this renders the wave system compatible with a wide variety of samples, such as serum, cell lysates, which often lead to clogging due to the big protein complexes or dense matrices. So our innovative microfluidics design integrates all the microfluidics within the disposable cartridge itself. So there are no microfluidic valves within the device core. So that's why it's also able to have a low maintenance requirement. And here it places large bore standard valves downstream of the instrument instead of having micro valves near the chip, as seen here. So this really allows for the ultra-fast transitions, as shown in this graph here, of up to 150 milliseconds for reliable determination of even the fastest off rates. So for example, we can measure the 10 per second off rate. And it makes the wave system highly compatible to a variety of samples, as mentioned before. So I hope this was able to give you a good introduction of what the wave system is based on. So really explaining our GCI technology and non-clog microfluidics. But now I really like to introduce as well our newest technology, so the waveRAPID, and how this is becoming a new way of really measuring kinetics. So as shown, for example, on the video before, where we're showing the GCI, traditional surface-based analysis techniques for determining kinetics first have a target that's captured on a sensor surface, and then there is an analyte flown over the sensor in multiple concentrations. And the binding is recorded in real time. So as seen here, so this is an example of multi-cycle kinetics, so traditional kinetics. And here we can see the sensorgram with the multiple concentrations. So in traditional kinetics, the interaction is probed by injections of a varying concentration, but with the same duration. So these injections have the same time. So as you can see in this plot here, the dilution series represented here in red, typically spanning 6 to 8 concentrations, are injected for the same duration. So this means that for each sample to be analyzed, 6 to 8 concentrations have to be prepared and injected. In order to determine the on and off rates of the interaction, both the measurement curve here in blue and the non-concentration shown here in red, as explained before, are plugged into the reaction equation describing the binding itself. Now, Creoptix has recently introduced a new way of measuring kinetics, and this is the waveRAPID. So RAPID stands for repeated analyte pulses of increasing duration. And here you see the need for the acronym. So this is a sensorgram of the waveRAPID, which, as you can see, it's slightly different than the one showing before because it's based on one single injection. So here the interaction is probed by injections of a varying duration, but from one single concentration. So therefore, we're just actually using one weld. So this is, as I was mentioned, as a typically measurement looks like. And here you can see that by making use of the fast transition times of the wave chip that I just explained before, one single sample and therefore one single concentration, as you can see the concentration is the same, is injected in repeated analyte pulses of increasing duration, just as the name states. So this really allows to mimic a concentration gradient directly on the sensor surface without the need for lengthy sample prep and serial dilutions. Again, in order to determine the on and off rates of the interaction, is similarly the measurement curve here shown in blue and also the non-concentrate function shown here in red are plugged into the reaction equation to describe the binding and be able to plot the kinetic rates. So ultimately, this means that waveRAPID allows to speed up and really streamline that interaction analysis process, allowing to save time and costs as more interactions are read in hours and not in days. As you can see here, it can be up almost to 10 times faster than traditional kinetics or multi-cycle kinetics. So this hit to lead screen is much, much faster. gets more insight. So there's a capacity to screen on a broad kinetic range, as you can see here, from picomolar to millimolar ranges as well. And there's also the ability to run more samples. So as I explained here, one sample really only requires one single weld. So this means that it really transforms a 96 weld plate into 96 data set. And also there is an ease of use because it really accelerates the kinetic screen. So you get all the kinetic information for the same time that you would take for a normal screening. Also using our direct kinetics tool based on the Google's AI TensorFlow, it allows for the fast one-click analysis, really focusing on evidence-based estimation to really reduce the fitting errors and allowing also the less expert users to effectively evaluate the data. So in conclusion, the GCI and waveRAPID technologies respond to a series of current challenges faced in drug discovery. For example, by applying the waveRAPID, there is a high increase in throughput versus the traditional kinetics, or multi-cycle kinetics, for example. Also, as it allows to read from one single weld, and even though getting all the kinetic information, it really allows to have several data points and the full kinetic and biophysical characterization. The non-clog microfluidics really reduces the worry of the clogging of the instruments and downtime, of course. And then both the waveRAPID and then also coupled with the direct kinetics tool really provides a much easier and transparent method to do the evaluation and the analysis. Also, very importantly, our wave systems, both the 2-channel and the 4-channel, as long as the waveRAPID, they all operate within the same dynamic range, providing the highest sensitivity in the market. So this really means that throughput does not come at the expense of sensitivity. So if you're interested in our technology and our wave system, feel free to visit our resource center, where you will be able to find several application notes, white papers, and also publications where our systems have been used, or feel free to contact us as well. So I'd like to thank you all for your attention. And now I'll pass the floor as well to Domainex, and we'll introduce Dr. Tom Mander, the CEO at Domainex, who will further introduce the CRO and how they are really supporting the drug discovery process. Thank you very much.
Thank you very much, Tatiana. Good morning, everybody from Cambridge here in the UK. So my name is Dr. Tom Mander, and I'm going to introduce you to Domainex, then hand over to my colleague here, Trevor Askwith, who's going to run through some of the applications that we've derived from our utilization of the Creoptix Wave technology at Domainex. So just to set the scene for you, it's my pleasure to introduce you to Domainex. So we're a drug discovery service company, as I say, based in Cambridge in the UK. We've been in existence now for 20 years. We were founded back in 2001, originally as a spin-out of a number of research institutes in London, particularly UCL, University College London, and ICR. Currently, we employ around 75 people at our Medicines Research Centre here, of which about 80% of our scientists, who are both chemists and biologists, hold PhDs. Despite the pandemic over the last couple of years, Domainex has been growing rapidly. So over the last couple of years, we've increased our revenues by about 80%. So that's obviously a testament to the work that we do here to support our clients in their drug discovery projects. We have a very strong reputation for delivering excellence. We're committed to delivering high quality data to our clients, and ultimately, of course, delivering them new medicines to take into patients. Our endeavors have been recognized by the industry. So we've picked up a number of awards over the years. Some of them are shown here. And so I would say, in summary for you, then Domainex is committed to working with our clients to deliver excellence in drug discovery, to support our clients move their drug discovery targets towards the clinic in an efficient and innovative manner. So that's the introduction for you on Domainex, and I'm going to hand over to Trevor now, who will give you some insight as to how we've been using the Wave Delta technology.
Yeah, thanks very much, Tom. So really what we want to do over the next few minutes is go through some details for a hit identification program that we've run using the Creoptix Wave and specifically the waveRAPID technology for the hit identification. So for a fragment screen. So the target we're interested in is a 50 kilodalton protein, and the project is looking at a protein-protein interaction. For this target, there are no small molecule inhibitors known, and we only have one positive control, which is a peptide derived from the partner protein. So for this project, alongside with the client, we decided that the best approach for hit identification was to run a fragment screen. And at Domainex, we have our own proprietary fragment screening collection of nearly 1,100 compounds. Alongside the assay work that we're doing, we've got X-ray crystallography running ongoing to identify crystal structures of the target. The initial stages for the assay development was obviously to immobilize the protein to the chip surface. And here we used a non-covalent NTA linkage using a his capture method. And this gave us really good levels of immobilization of around about 10,000 picograms per millimeter squared. And this is the high level of immobilization we would want to enable us to get accurate data for a fragment screen. On the left-hand side at the bottom here, you can see one of the traces that we obtained using the positive control peptide. And this demonstrated that we had binding affinities in line with the other biophysical data for this peptide. So moving on in terms of the fragment screen, what I'd like to do is highlight the figure on the right-hand side, which shows how you would run a traditional kind of chip-based fragment screen in red and compare that to how we run these fragment screens using the waveRAPID. So in a normal chip-based system and in parallel with kind of other hit identification methods, you'd normally want to run your primary screen, which would be a single concentration screen. You would then rank this on the basis of binding levels, and then you'd run a secondary screen, maybe at a couple of different concentrations, rank this again based on affinities, and then you would run full concentration response curves, and then you can rank based on the kinetic data that you had obtained. Using the waveRAPID screening technology, we can bypass many of these steps now. And so we run the primary screen, and this is using waveRAPID, so we're able to obtain the full kinetic data straight out of the primary screen. So the compounds are put in at a single concentration of 250 micromolar, and using the waveRAPID screening method, we're able to obtain full kinetic data out at the other end. And this is what we ran for this project. As I said, we ran the full fragment collection of just under 1,100 compounds. These were run at a single concentration using the waveRAPID mode, and the entire screen was completed with 4 overnight runs of about 15 hours. And as I mentioned, what you're getting out at the other end is full kinetic data, affinity data, and this is all obtained in the primary screen. We're able to filter the data on the basis of the Rmax, so where we have confidence that there's good binding there, we're able to filter these data out. And at the bottom, we're able to obtain the on and off rate plots directly on the basis of that primary screen. In this example, we went forward and the screen was a success. We were able to obtain 30 fragments that had binding affinities of below 150 micromolar. And just to confirm the method, we went on and ran these in a traditional kinetic mode. And so we compared the rapid data on the left-hand side to the traditional data on the right for each of these fragment hits, the couple of examples shown on the screen. And there's excellent the data lines up very well next to each other. All the hits were within threefold. Most of the hits were much closer than that, and within twofold in terms of the binding affinity between each other. Here at Domainex, we've got a very thorough biophysics platform, and so whenever we run a fragment screen, we always look to confirm the fragment hits using an orthogonal method. In this instance, we took the 30 hits that we obtained from the primary screen and ran these in a nano-DSF method. So this is a label-free thermal shift assay. These were run at a single concentration in duplicate, and 5 of these 30 compounds were confirmed as binders in the nano-DSF. And you can see example traces on the right-hand side. At the top, you have the positive control, and then on the lower down, you can see the shifts obtained from the fragments that confirm binding. As I mentioned, we like to have X-ray crystallography running alongside all our fragment screens, and we have in-house expertise within crystallography. And these hits were taken through into X-ray crystallography, and we were able to obtain high-resolution crystal structures for 2 of these compounds. And this enables fragment expansion work, which was structurally enabled. So moving forward into the hit expansion phase, and this also exemplifies how beneficial the waveRAPID screening technology can be within this phase of the drug discovery program. So what we like to do whenever we obtain fragment hits, the first stage is to look for similar chemical matter that's either in-house or commercially available. And this phase of it requires, again, very rapid, very quick assay screening. So for all of these compounds, they go into the Creoptix Wave Delta, and they're all run in the waveRAPID mode. So again, in the same time frame that you would normally obtain single concentration data, we're able to obtain and the association, the dissociation plot. And this highlights the advantage of being able to obtain the kinetic data straight in the primary screen has. So here I've highlighted 2 example compound series that came out of the original fragment screen. Series 1, we're getting an increase in the association rate, and then series 2, we're getting a large increase in the dissociation rate, so decrease in the dissociation rate. So the compounds that are coming through from series 2, the affinity is driven by a reduction in the dissociation, and this is the series that we can focus upon with our chemistry efforts. We make early on in the program, which really increases SAR. Here's another advantage that the use of the waveRAPID technology has within screening, especially in the early phases of screening, and that's being able to run parallel screening in the different channels. So we have the Creoptix Wave Delta, which has 4 different channels. One of the channels is a reference channel, and then we're able to immobilize 3 different targets to the 3 different channels. We're then able to screen the compounds coming through in parallel, and in one overnight run, we can get 300 compounds run through to obtain full kinetic parameters. That's the binding affinities, the association, the dissociation rate against not just the target of interest in the first channel, but also selectivity targets, for example, in channel 2 and channel 3. We're able to obtain these data straight off. The other position this can be an advantage for is when you're looking at early stage projects, and maybe you've got different constructs that are going through. Maybe you've got a wild type construct, and then you've got a couple of different constructs that you're interested in with respect to crystallography. We can do this as well, where we would immobilize a wild type construct in one of the channels, and then 2 putative crystallography constructs in the other channels. And that means that rather than going back and backfilling some of these data, we get these data straight out with the primary screen that we're running. So in summary, we've incorporated the waveRAPID Creoptix Wave and the waveRAPID screening technology into the platforms that we run at Domainex. It replaces the need for single concentration screening. This minimizes compound handling time, and because you don't need to prepare concentration response curves as well up front, this also minimizes handling time. It accelerates assay validation, it's not something I've spoken about an awful lot, but the first stage is when you're validating the immobilization of your protein, is to put some positive control tool compounds through, potentially peptides, to validate the immobilization of your target. The fact that the waveRAPID screening is so much quicker than the traditional method, it means that you can validate the assay faster. And if there's an issue with the immobilization or you're getting maybe non-specific binding, it's much quicker to go back and rectify these issues, which means that you're running into your screening much faster. As I've highlighted, it accelerates hit identification. We're able to obtain full kinetic data with the primary screen, which means we're less likely to miss compounds, and we can prioritize the compounds that are more interesting. It accelerates routine SAR. As I said, we're not running single concentration screening, we're not missing compounds, and we can really focus on the chemical series that are coming through that are most promising. And all in all, this reduces data turnaround time, and increases the number of design make test cycles that you can run in the same time. In conclusion, summary, I'd just like to thank the scientists using the system at Domainex, and here are some of them listed below, and obviously Creoptix as well, who are really very helpful, not just in terms of the technical input they have, so especially with Ted, who's one of the technical specialists, and Casper, who was also here when we installed the system, who's one of the inventors behind the technology. Now's a good time to hand back, see if there are any questions.
So thank you.